This is below Lake Mead. The supply, it goes up and down. You can see the two big peaks in the 80s and late 90s. Those were the El Nino years, the massive rainfall years. But the average is around 50 million acre feet. But the demand is already exceeding the rate of delivery of new water. And this has gotten only worse since this chart was created. And right now there's a crisis on the Colorado River. So what's freshwater used for? Primarily agriculture, and 80% of agriculture is for animal feed. So, approximately more than 50% of irrigation water in the United States is used for animal feed. And about 70% of the irrigation water near me in Imperial Valley only gets water from the Colorado River. It only has an inch or so of annual rainfall or less, and it's a desert. And it's good soil, and they grow alfalfa there. And so there's a massive use of water by humans to grow fodder for our animals. What about fuels? In the future, we're continuing to have a demand. Human population's growing. The projections are we'll need more energy. Not only, we can't only use electricity or solar or wind, and biofuels are one of the wedges of the options into the future. You can see in this chart, biofuels are projected to grow into the future. Now, where will all that biomass come from? If you look out 2030 and beyond at the wedge that's biofuels in this chart, you actually convert that through the terrestrial system and look at how much biomass we'd have to grow of, let's say, soy or any of these other crops that can be even palm oil. What you'll find is that there's not enough land and water to actually produce the biomass to create those biofuels, and we just will run out. And so, what are the options? Are we going to not use biofuels, or is there another option? And where will the biomass come from with declining water and arable land resources? So I'm going to switch, into kind of my research domain. And I'm going to present information on 3 levels of work that I've done. On the first, I've done work, as I mentioned before, the global oceans, collecting data on the oceans, on optics in the ocean and the ecological processes of the ocean, including the biomass of the ocean, the chlorophyll, the carbon flux out of the ocean into the sediments or into the deep ocean, and the exchange of oxygen and CO2 with the ocean. And on the left is a model we've run based on the global ocean net community, global Southern Ocean net community production. Net community production is carbon that's exported from the surface into the interior of the ocean, fundamentally removing carbon from the atmosphere. Southern Ocean does a lot of that. And we've done, I won't go into the details of how we did that, but on the right, the annual cycle of that process is compared to the annual cycle of oxygen in the atmosphere. And we've used oxygen in the atmosphere to constrain our forward satellite model. So we measure optics of the ocean, measure chlorophyll in the ocean, measure primary production. We run a model forward with the satellite, but it's unconstrained. The oxygen in the atmosphere is an independent constraint of that. And again, I'm not going to go into details of that, but this is global scale work. Also, I've worked on biophysical models of growth based on the absorption and quantum yield. Light is absorbed by plants and algae, and the photosynthetic quantum yield is the transfer function that turns photon flux into carbon fixation or oxygen evolution. It's called the quantum yield of photosynthesis. And third, I'll talk about metabolic models that we use to elucidate a novel photorespiratory stress pathway that was quite a surprise. It appears that the chloroplast and mitochondria in diatom are very tightly coupled in ways that were not really appreciated from work primarily with plant photosynthesis. So in terms of biophysical models of photosynthesis, why do we care about modeling these things? Well, I showed we apply to global carbon cycle understanding, but also if you're going to use some organism like algae or corn for economic production of an agricultural product, you're going to have to be able to predict its outcome. And there's a tremendous amount of data about, as all of you know much better than me, agronomy and knowledge about, yields of corn and alfalfa and wheat and sorghum and everything else, soybeans. But there's very little knowledge about algae. And so I've worked on laboratory studies to try to understand the growth regulation of algae and how to predict their outcomes. And the figures there on the right, well, on the left, it shows the light on one axis and temperature on the other axis. And it shows basically the three-dimensional matrix of the light temperature co-limitation matrix for the growth of one organism one microalga. And on the right is just a plot of the predicted growth rate versus the observed growth rate. So the point is that we can take these fundamental biophysical parameters, absorption and quantum yield, photosynthesis, and we create models using that. And so in terms of how we would apply this in terms of, let's say, industrial biotechnology for algae to try to find strains that would be commercially viable, there's tens of thousands, if not hundreds of thousands or millions of strains of algae in the world, lakes, ponds, oceans, and everywhere. And so you need to do some prospecting, some screening. And, if things are promising, then you could go to more detailed experiments. And then the detailed experiments is you've got to be able to have yields that you want, the biochemistry that you want as an outcome. And furthermore, you've got to be able to make predictions so that whatever you're going to grow, you can predict its outcome in terms of your production system. If all that's working out well, then you have a potential commercial strain. And so this is the kind of work that I get involved in and with the Department of Energy Research and some of the collaborations in industry. In terms of the actual details of the model, primary production can be by carbon of the absorption, the APH, so the A star pH, chlorophyll in the second equation there, the red term. And so the photostatic quantum yield, the phi, photosynthesis, and A star pH, these are functions of light, E, nutrients, N, and temperature, T. And that's what I call the co-limitation matrix. And there's been there's a classic concept of the law of the minimum, whatever the minimum limit is. But it turns out that algae have really a plastic phenotypic expression, and they adapt within a co-limitation matrix. And most experiments have done with a single limitation. So it ends up being, a little bit underspecified if you don't actually explore this in the co-limitation matrix. So if you look at, let's say, the green oval is an algae cell, you've got the genome, obviously, the DNA. That green oval is an algae cell. It's another and then you've got the metabolome. Oils are produced, carbohydrates are produced, so forth and so on. All of these, this entire system within a cell is regulated by light, temperature, and nutrients. Now, it turns out that in a production system, we can manage the nutrient, we can control the pH, we can control the salts and other things. Two things that we don't have as much control over are light and temperature, because you might be growing, let's say, in Prairie View, where it's cold in the winter sometimes, or freezing last winter, or very hot in the summer. And so your crops, you're going to want to have crops that can manage it across the light and temperature variability that you've got. So that's the way I explore my work. And I'm going to give two examples of models, and I'm going to go through some of the slides quickly so we can get on toward the end and the conclusions. So if I don't go into the details of the next coming up slides, bear with me. So in the laboratory, we've studied a number of different strains of algae. They're listed at the top. I won't bother to read that. And we've especially looked at light and temperature as regulators. And you can see the kind of growth systems we established in the lab on the right. On the left is an image of a Thalassocyrus pseudonana diatom. And in particular, Thalassocyrus pseudonana and Phaeodactylum tricornutum are model organisms. The genomes have been known and documented very early. There's a lot of physiological data on those. So we tend to work on model organisms. This is a set of equations that we use, and we can measure the growth rate, the mu, we can measure the chlorophyll and the carbon, we can measure the absorption coefficient, we can measure the light. What we can't measure effectively is the quantum yield of photosynthesis. We can also get estimates of respiration. And so basically we can solve this equation for the photosynthetic quantum yield. If you look down at the bottom, you see the phi mu, that's the growth, that's the quantum yield of photosynthesis for growth. And we express it as a function, in this case, of light. We can also do it as a function of light and temperature. It's a little bit more complicated. Here's, as you see in the, not the lowest equation, but the second lowest, the chlorophyll to carbon ratio. We model it with that function. Here's a chlorophyll to carbon ratio and the function we use to fit it. We could fit the data very well. Here's the absorption coefficient per unit of chlorophyll at one wavelength. Well, the three different wavelengths, the 440 is blue, the red is 660, and 680 is the red is 680. I'm sorry, the red is 660. What did I say? The green is 680, which is the chlorophyll peak. 440 is the chlorophyll peak in the blue. But the point is we can do a good prediction of the absorption for chlorophyll. Here's our estimates of respiration. There's not much respiration data. And so we've taken data from the literature that don't tend to go out to very high irradiances where you end up having photoinhibition. So we kind of speculate what the function of that would be as you go out to high intensities. Here's the quantum yield of growth and the equations that we use. And what you can see is in red, that's the gross photosynthesis. In other words, that doesn't include respiration. The blue is net and the fit to those two curves. So you can see that we're doing a good job of predicting the quantum yield of photosynthesis. And when we put it all together, we can predict, in red, the gross photosynthesis that doesn't include respiration. The blue is the net photosynthesis. You can see the photoinhibition at high intensities. And in green is the estimated respiration rate. And again, if you look at the modeled growth versus measured growth, we do well on the left. And if you look at the modeled quantum yield for growth versus the measured or estimated based on laboratory data, again, we have a good correlation there with a slope of almost 1-to-1 in both of those. So now if you and say, okay, well, how does this apply to, again, the temperature and light gradient of an organism, in this case, Denali tertialecta. The one I showed earlier was actually for an Antarctic diet, an Antarctic mesophyte. But you can see that there's a three-dimensional matrix of light and temperature regulation of the growth rate. And this is what we have to understand in order to optimize any sort of application of algae to a viable agricultural model. We have to be able to have a good prediction based, as you can see on the right, prediction versus measured is good. So I'm pretty confident we have a framework to model microalgae. We haven't really scaled these kinds of things up at massive scale, but that's what we're looking at. So the conclusions is that light, nutrient, temperature are fundamental variables that regulate the growth, and in particular key biophysical variables, the quantum yield of photosynthesis and the absorption of chlorophyll are functions of light, nutrient, and temperature, and we're able to model those. And I believe that this is going to be important for economic viability in the end. Now I'm going to go down subcellular into a diatom. And so what I was showing you before was if you take the whole cell and you look at its growth rates, you look at its bulk carbon, its bulk chlorophyll, its bulk absorption, and so forth, you can make a model of the overall growth of the total carbon in the organism. Now, it turns out inside the cell, there's a vast number of metabolic processes going on, and it's important to try to understand those as well. Now, the diatom Phaeodactylum tricornutum has an interesting attribute. Most algae will saturate at maybe 10 or 20% full sunlight intensities, and then at higher intensities, they'll have serious photoinhibition. Usually, if you're more than half of full sunlight, most microalgae will be so photoinhibited, their growth will be dramatically suppressed, or even it'll kill them if you get to too high intensity. But Phaeodactylum tricornutum can continue to grow, not at increased rates, but it saturates and then it doesn't photoinhibit. I'll show you some data in a minute. And if you look at, I'm going to kind of give you the conclusion first, because the details of the metabolic model are kind of hairy, and I'm not going to go into detail. I'll just go through some slides quickly. But on the left is a classic model of what is a plant cell. So those of you that are agricultural experts and studied plant physiology, there's mitochondria and chloroplast inside of plant cells, and they're separate organelles. And actually, there's an ultrastructural image on the left, and they're separated. But on the right, what you can see is that the chloroplast and the mitochondria are in really tight conjunction with, and zooming in, there's a lot of interplay in the membranes of the two, and these are obviously chloroplast and mitochondria are derivatives of bacteria from an evolutionary source, from the massive lateral transfer of genes by bacteria or cyanobacteria being incorporated into higher organisms. And so these have their own discrete membranes, they have their own discrete DNA, and they have their own discrete functions. But in the diatom, they're really tightly coupled. And so the classic model that I just showed you for the plant is you got the chloroplast and the mitochondria, they're separated, excess oxidative stress gets managed by something called a peroxisome. I'm not going to go into detail of all the metabolic pathways, but what we're seeing in the Phaeodactylum tricornutum is that they're tightly coupled, and some of what would otherwise be oxidative stress and just be dissipated by a peroxisome ends up driving a secondary pathway for catabolic processes. And so what do? We've got what's called a genome scale model, and that requires the knowledge of the full genome and there's no plant or animal that we have full model of all the metabolic processes. We have full genomes, but not necessarily full metabolic processes, but there's a large matrix of information known about many organisms, this bacteria more than others, yeast, but in some cases algae. And so we put that into the system, and these are called constraint-based or COBRA genome scale models. So what's the constraint? You can constrain it by the conservation of mass and conservation of energy. If you have a heterotrophic organism, you constrain it by the conservation of mass. How much sugar or organic matter did you give it? If you have a photosynthetic organism, you've got to constrain it by how many photons are absorbed too, the energy and the nutrients. Well, in the past, people did a bad job of constraining based on the light absorbed, but fundamentally, that's what I do. I measure on the upper left here is the absorption coefficient of the Phaeodactylum at different light levels. And in the bottom, you can see the photosynthetic rate as a function of the radiance. Now, the radiance is going all the way out to about 2,000 micromoles. That's equivalent to about full sunlight. So what you can see is, although it saturates around 200, 300 micromole in the lower left here, the photosynthesis versus the radiance curve on the lower left saturates and doesn't inhibit at high light. And when you put these actual photon flux information into the GEM model with a conservation of energy constraint, we're able to solve for things inside the system. And I'm not going to go into all these details here, but I'm just going to come down to, let's say, the conclusion here is that we've done this metabolic modeling. And what we found is by using the quantum flux and photosynthetic rate accurately in a metabolic model, we can make constraints that end up giving us a conclusion that the direct metabolic interaction between chloroplasts and mitochondria. And our conclusion at the bottom here is that this is in part what may lead to the successful acclimation of Phaeodactylum tricornutum under very high light conditions. And so, these are very technical details, metabolic details, but certainly agricultural experts, people in all sorts of universities like your university and other agricultural universities do study the photosynthetic performance of all different kinds of crop plants. And we're doing this for algae. Now, a good question would be, why algae? And Professor Levine's talk in a couple of weeks will be, why not algae? One why not that I'd like to point out is that if you look on the upper left here, land plants are derived out of the strain, the evolutionary pathway of green algae, the chlorophyta, and they branched off some hundreds of millions of years ago. But if you look at the full diversity of life, which this diagram represents, you can see on the upper right, dinoflagellates, on the right, the diatoms, the basilaria phyta, the brown algae like kelps. You can see animals over on the lower left. And when I give a talk, if you look at this diversity, you go, wow, that's amazing. If you track animals back down that purple chain on the lower left, the humans are sort of there evolving late in the evolutionary cycle. But it turns out that we're more closely related to a flatworm than a green algae is related to a red algae. And that's a take-home thing that shocks people. So in other words, red algae and green algae aren't actually related to each other. They're called algae, but they're not related in the same way that we're related to a flatworm, for example. And so what does that mean? What that means is over several billion years of evolution, algae have been able to, they've evolved in very different niches in very different ways. And we can expect that there's very diverse metabolic pathways that are undiscovered. The prospecting of algae and their metabolic pathways to create biochemicals that may be useful industrially or economically or agriculturally is only just beginning. So instead of just only looking over here in this upper left corner in the chlorophyta, with algae, we can start to explore a vast domain of evolutionary metabolic performance and perhaps biochemicals that we never imagined before. And I think that's something that any young student might get inspired by and say, hey, I want to go discover what's over there in the unexplored part of the natural realm. So for sustainability, we can't separate food, fuel, water, land, nutrients, greenhouse gases, socioeconomics, and policy. And I know that you guys are really looking at that closely. And, food, energy, water are all tied together and linked to hunger and socioeconomic development. Terrestrial crops lead to topsoil erosion, deforestation. There's limited arable land, there's limited water, and the direct and indirect land use change are issues. Climate change is an issue, biodiversity and habitat loss to agriculture. And it's largely related to feeding our animals, humans, cows, chigs, and I put pigs in caps because I love this quote from George Orwell. The creatures outside looked from pig to man and from man to pig. It was impossible to say which was which. Now, I'm not calling it issue they're pigs or myself a pig, but Orwell had an interesting way of looking at the world in a kind of a cynical way. This was in National Geographic back in 2007. There's no magic bullet fuel crop that can solve our energy woes without harming the environment, says virtually every scientist studying the issue. But most say that algae comes closer than any other plant. And algae can also be a magic bullet protein and sustainability crop, not just fuel. And I'm going to go into a little bit of detail of just some yields and some metrics about algae biochemical. So if you look at just the protein in algae down at the bottom, you can see they're all more 50% or more for the most part, far more than corn, more than soybeans, more than milk. They're similar to fish and meat. So algae have really high protein content, and protein is really a key nutritional element for agriculture, obviously. But if you look at the yields on the top, for corn, soybeans versus algae, the yields of tons per acre and the tons of protein per acre, I mean, you can see that algae are going to produce 10 to 20 or more times protein per area per time than any crop. Now imagine doing that without losing topsoil, without wasting any agricultural nutrients or fertilizer, without deforesting, without, and even doing it on saline water. So if you look at, for example, potential yields, this was for Imperial Valley, but and this is not considering the cost of production. This is just the value of the crop. The crop could be worth 20 or 30 times more per acre than an agricultural crop like alfalfa, and so I think that while the cost of producing algae is high, the yields are so high, we have to really plug that into the economics. And so they have exceptionally high yields. The yields imply that fuel and animal feed for 10 billion humans could be produced on half the land area currently used for agriculture. They use non-agricultural land, so the higher quality lands could be allocated for tree carbon storage and big red caps. We could reverse climate change by sucking CO2 out of the atmosphere this way. And I think there's a better way to do it than sinking seaweeds in the ocean. Algae can grow on seawater or saline water. We've thought before they need the point source CO2. It turns out that some of my colleagues are now getting algae yields very high on direct air capture. There's minimal waste of nutrients. They'll reduce greenhouse gases. So the theoretical merit of algae is not disputed, and there's been a lot of progress. So this is my vision of sort of what we need to do. In the beginning, I said we need to do strain isolation. We have to do evaluation. Once you've done lab work like I do, then we need to interface to engineers, agricultural engineers, agronomists, and look at how do we design the system that would optimize the biological potential of algae at the lowest cost. How do we iterate on that and collaborate? And I think this is the proposed breakthrough opportunity, is to actually combine the agricultural, the engineering, and the biological in an effective way. And I think there's a lot of opportunity for bringing in students across that whole spectrum of disciplines to really explore how to do this. There's been significant progress in strain isolation, genome-enabled systems biology. It gives us a chance to really understand what's going on subcellularly. Efficiency will allow reallocation of quality land for tree carbon storage in a distant future, but I think we shouldn't lose track of that. And, this was a pitch I made to Gates Bioenergy, Breakthrough Energy Ventures, BEV. And we had a team that I put up. I should have gotten rid of that last bullet because I'm not proposing a team to you. Here's a picture of an algae farm in Imperial Valley, about 120 miles to the west, to the east of me. It's a desert. They get about an inch of rain a year. But all the water that goes in there to mostly grow alfalfa or other field crops comes from the Colorado River. It's all irrigated, beautiful soil. It's all perfectly flat. It's a great place to grow algae. If you look at how would we get there, there's something called the progress ratio or industrial economies of scale. Right now, if you're looking at just fuel, we're somewhere with algae, somewhere like $1,000 or a couple $1,000 a barrel of fuel, far too expensive up at the upper left of that graph. And we need to get to larger scale. And so if we don't get to scale, I don't think we're ever going to bring the cost down. And we're going to have to be able to put the investments in, take the risk, accept some failures, and get along. Right now, if you can see the arrow in the middle toward fish meal, that's a higher value. meal or cornmeal or soy oil or corn oil or petroleum. And I'm not going to go into too much more detail here, but if we don't get on this scaling up and learning curve, we're not going to bring the cost down, and that's a reality. And we're stuck way at the top still, and I'm advocating for more investment. Here's one of the investments has been first, the Defense Advanced Research Projects Administration, DARPA, funded this facility in Kauai. I was a subcontractor with General Atomics on this project 10 years ago. General Atomics didn't want to pursue the commercialization, but the team and the lead chemical engineer, Dave Hazelbeck, rolled this facility and the team out into a company called Global Algae. And you can see there's a Facebook video if you want to go take a look down at the bottom. But this is a small facility, but you see the larger ponds. I think the largest ponds are like 5 acres, but they've got a design now for 25-acre ponds, and you scale them up. And if you had hundreds of 25-acre ponds, you could make a lot of algae. So in conclusion, this is my main oceanographic and my main career has been going to sea on big ships in the middle of the ocean and putting fancy instruments under the water on the end of the cable. That instrument on the right is about $500,000 of electronic junk that measures all sorts of things about light and photosynthesis underwater. And but I also have done lab work, and I'm collaborating with industrialists to try to push the algae commercialization opportunities forward. So thank you very much, and happy to answer any questions. And I think we still have a few minutes. I hope that wasn't too fast, and sorry to have started late, but you got my slides, and I would welcome, I think my email's on the first slide, and I'd welcome emails or contact from any of you, and I hope to meet you all in person soon. Thank you.
Well, thank you, Craig. Thank you, Dr. Mitchell, for this wonderful presentation. I think before we allow the questions and answer, I'd like to give the Dr. D'Souza a minute. I think he wants to introduce one of our visitors, and then we can resume.
Thank you very much. As promised, I'm going to deliver a special guest. So we're going to break away from our regularly scheduled broadcast for a special announcement that Dr. Myers himself is here visiting for the day, well, for a few hours anyway. So I enticed him to come and speak to you for a few minutes. Dr. Mitchell, great speech. Really loved what you had to say. And so without further ado, drum roll, please. Dr. Myers. So
Greg, we can stay after 12, no problem.
Yeah, I'm fine too. I'll hang in here.
All right. Good to see everybody. Yeah, I saw, albeit virtually.
Good to see you too.
To see everything that's happening, and just had a chance to visit with Dean D'Souza and hear that things are still moving forward. In fact, I understand that World Food Day is coming up, Mata, and I understand that you're moving that forward. So it's great to hear that that's happening. The presentation today, I think, is very relevant in this whole area of food security and looking at another food source, a protein source, which is very, very important, particularly in these times. And Prairie View is right on the cutting edge. I mean, when you look at what's happening, and I see what's happening in the world, and many of us, it doesn't take much for us to look into in the news and see what's going on. And Prairie View, Texas, is right here. We're in the cutting edge of it. A lot of what we're seeing that's happening on the border, and people coming and trying to meet their needs and whatever, is here in Texas. That's where it's at. That's where the action is. And Prairie View, I believe, still fits into or sits in a very important position in looking at what the needs are and how to fit those needs. And Prairie View is very well positioned for that, with the Food Security Center now, and with the teaching that we do here, and then the relationship that we have with A&M. There's some opportunities, there's tremendous opportunities. And so it's good to see what's happening. It's good to see that you're here. And even in the midst of what we're doing, what we're having with this pandemic, Prairie View has not stopped. Prairie View is still moving forward. And that is great to see. Keep staying there. Keep doing what you're doing. I'm going to get a chance to walk around a little bit. As many of you know, I served as a chaplain for the football team, and the football team found out I'm here. So I've got to meet about 60 football players here in about 45 minutes to get them ready for the football game tonight that's going to be broadcast. But again, it's good to see everybody. Let me just say this. I gave a message at our church a few weeks ago. And the verse said in Ezekiel 22:30, it said, I was looking for somebody to stand in the gap so I wouldn't have to destroy the land. And I couldn't find anybody. And the land got destroyed. Prairie View, I believe that that is a challenge for this university and all the scientists. Are you standing in the gap? Are you at that point where there are issues and where there are needs? Prairie View is the place where you stand in the gap, where folks are hungry. You stand in the gap to try to meet that need, where there needs a need of education. People need to learn what's going on. Prairie View, you're the one that stands in the gap. You're the one that sits in that place so that the land doesn't get destroyed, that the people's needs will get met. There are a lot of other universities doing a lot of other things, but Prairie View's got a very unique position, I believe, to stand in that gap. And so, again, many of you know I can stand on a soapbox and talk all day, but I really believe that. I really believe that there is a very important place that Prairie View sits, and there are opportunities there. And so, again, it's good to see all of you, as albeit virtually. I'll be walking around here. The dean is going to take me around a little bit to hopefully see you. Again, probably all masked up and whatnot, but that's just the nature of where we're at right now. But again, it's good to see everybody. It's good to see the campus. It's good to see things are moving forward. And again, I am now serving, I have the title of emeritus professor, so I'm still associated with Prairie View. So I can still be tapped into. If I can help out in any way, given, again, my position and where I'm at now, let me know. So thanks again, and good to see everybody.
Are you coming to CRC, Dr. Myers?
Dean D'Souza said he's going to try to get me over there. So hopefully we'll get there.
Good.
Yes, sir.
Dr. Atai will keep him here. We're not letting him out over there. So.
I'll take him for lunch.
Well, we tried to entice him to stay for lunch, but that's not possible. I will take him around ag business, 4th floor and 3rd floor. Then we'll move on to CARC. Anyone who wants to meet him, be in the front lobby of CARC. And then finally CEP, in the lobby of CEP. And then I'll take him for a windshield tour of the farm. So we're going to try, but I learned something today. Prairie View must stand in the gap. So thanks once again for that great message, the powerful message. And Dr. Mitchell, didn't mean to steal your thunder, but Dr. Myers, now retired, was a longtime professor here at Prairie View A&M University in our college, and he was the endowed professor of food security. So he's now emeritus professor. So it's not every day that we have a visiting dignitary. Unfortunately, it happened to be in this time slot.
Well, I love his enthusiasm and also the challenge he presented.
Absolutely.
There's a gap, and it's up to us. I'm also emeritus. I hate to say how many years I spent at the University of California. I'm still active there, but it's the students that we teach and the students that they then teach, and that's going to fill that gap. And the educational system is so fundamental. And I love the enthusiasm.
Great. Thanks, colleague.
Thank you.
I'll take him around.
Thank you, Dean. Craig, I think we're going to open the floor for questions and answers. I just want to let you know that this is just the beginning, because Dr. Mitchell is a historian, and he will be visiting us in person, and we will be hopefully furthering our collaboration even further, so with other opportunities. So this is just breaking the ice here. So we have time for a few questions. If you can go ahead. Anybody has a question or comment? Maybe our incoming Associate Director of Research, if you have any question or comments, Dr.
Yes.
Erdogan.
Yes, first, Dr. Mitchell, thank you so much. This is Erdogan Memoli. I'm a professor here at Mississippi State, but I'm honored to be the future executive director of the CARC there at Prairie View. Really enjoyed your presentation, and I'm glad that you touched on to genome biology. And I was curious if you could talk a little bit more about how metabolic pathways can be engineered using gene editing on those promising strains that will adapt to temperature, light, and all those other parameters that can be more useful.
Yeah, well, I'm not a gene editor or molecular biologist. I'm a physiologist and ecologist, but I view the future as vast. Obviously, there's social concerns and potential risks that have to be mitigated, but then there's vast opportunity. It's pretty clear that almost the entire agricultural domain, whether it's crops or animals, are benefiting in terms of yields and all sorts of resistance to disease and pests and so forth through molecular biology interventions. In terms of microalgae, we're only just beginning. And as I showed you, that evolutionary fan of the evolutionary diversity of all of life, algae are out on 5 different trajectories on that fan of all life on Earth. And so there's a vast genomic diversity, which must also represent a vast metabolic diversity that's unexplored. So we actually don't know much about it. There's very few, most of the organisms that have been, even the genome fully documented in terms of microalgae, there's not that very many of them. And in terms of detailed metabolic understanding, it's just barely starting. So I consider the opportunity huge, but in terms of speculating exactly what it might be, I don't know. I think that I think we'll discover it as we go along. But a huge advantage of microalgae is that the ones that are commercially viable can double their biomass or even triple their biomass in a day. Their rates of growth are very fast. So from a point of view of discovery of what you could do with an organism through, let's say, gene editing, if you want to do a soy or corn, good luck if you get 2 crops a year, maybe 3 in Hawaii or something like that. With algae, you get 3 divisions in a day. And so the accelerated pace at which we'll be able to discover these metabolic pathways, whether it's just documenting the metabolic pathways or modifying them and seeing what the modifications do, the pace will be very fast with these microorganisms, similar to bacteria and yeast. And so I think that that's a vast opportunity unexplored yet. I mean, some labs are doing it and some companies have been doing that, but I think it's just beginning. Thanks.
Well, thank you so much. I really appreciate it. Years ago, I studied microbiology at the University of Wisconsin-Madison. And it's so inspiring to hear your updates. And you're right. I think opportunities for future students are just amazing. Finally, I'm co-organizing cattle, goat, and sheep session at the International Plant and Animal Genome Conference in San Diego in January. You sound like a great guest speaker there as well. So I'll reach out to you later.
Okay. Well, when is, just you got my email. Dr. Ferris has my email, so I welcome your email. Thank you.
Great. Thanks.
Thank you, Dr. Mimley. And by the way, we're in discussion of strengthening our collaboration in algae. So I will update you on that discussion. And Greg, Dr. Mimley should be in that discussion because he will be the person who's going to be helping us with that.
Yeah, so, well, anyone you CC, if you think I should be CCing, I don't want to bother too many people, but I look forward to further developing more relationships there. Thanks.
Well, given the time span, we have one time for one more quick question, please. Anybody who would like to?
Can I ask?
Sure. Go ahead, Dr. Kabroum.
So I'm plant physiologist. I have been studying photosynthesis long time ago, but now I am moved to plant development. So today's your seminar, it was very interesting because I never thought that algae are that productive. I have seen them being studied, for example, at Texas A&M by chemists or biochemists, not plant biologists. So I have two questions. One is about this tight coupling of mitochondria and chloroplast. How that happens in algae? And the second is that you said protein production in algae is very high from what you showed the data. How is it calculated or, for example, harvested and things like that? Can you explain those?
Yeah, well, in the case of the diatom evolution, so diatoms, and that's the model organism that I was presenting about the coupling between the chloroplast and mitochondria. So in the case of, let's say, green algae and higher plants, you know, the chloroplast was derived from a cyanobacteria into some sort of organism, and then it evolved along those lines. And in the case of the diatoms, there were more than one massive gene transfer. So probably more like some sort of animal ate the original cyanobacteria. That entire thing got incorporated into another animal. And so there's multiple layers of membranes inside of the diatoms that's very different than it is in the higher plants. And you know, exactly how that leads to the evolution of this tight coupling, you know, I'm not going to speculate because I don't know. But the point is that what we see from an ultrastructural point of view is we see this relatively surprising coupling. And then when we run the metabolic model, it looks like excess electrons and that would otherwise probably cause reactive oxygen problems in the chloroplast somehow get shunted into a metabolic path in the mitochondria. And we haven't necessarily proved that, but that's what the model shows. And so, you know, that's probably not a great answer to your question, but as a physiologist, I don't run the models, and I'm also not a metabolic engineer. I collaborated with co-author Bernard Paulson, who's the founder of a constraint-based whole genome models at the UCSD, was a co-author with his student. In terms of the productivity and the yields, you know, we harvest the biomass out of the water, concentrate it down, dry it, and the yields are based similar to plant biomass yields, is how much protein per dry mass and how much dry mass produced per unit water, per unit nutrient, per unit area, per unit time. And so these are units that are comparable to agricultural measurement units, and the yields are very high. What we haven't been able to prove is that we can produce the biomass inexpensively. And so there's still a challenge to bring the cost down. But the fundamental biochemical reality is that algae are anywhere from 30 to 70% protein, and typically about 50%. So their protein is high. Some of them have a lot of oil. Some have a lot of carbohydrates, but I was showing the ones that had high protein. Yeah, thanks.
Thank you very much. I think we are going to give you, Greg, the last word here. But before we go, I just want to thank everybody for attending, and especially our students who really, I see them active in the chat. So thank you for attending, and please, our faculty, encourage your students to attend, because I mean, this type of opportunity doesn't go unnoticed. I mean, we need our students to benefit from experienced people such as our speaker today, Dr. Craig Mitchell. So, Craig, can you please go ahead and give us closing comments?
Yeah. Well, thanks so much for inviting me. And sorry I couldn't be there in person, but I saw your open calendar date and I jumped on the open one right away. I do plan to visit at some point, and I'll be in touch with Dr. Ferris regarding that. My email is in the first slide of the presentation. Any student, any faculty member, any staff member, any of you want to contact me and learn more, I'd be happy to correspond and give you some links. Dr. Ferris mentioned that there's going to be we're in a discussion about something called the Algae Technology Education Consortium. There's 5 or 6 Texas colleges and universities already part of ATEC. It's funded by the Department of Energy. And there's another half dozen around the nation. And so we're looking at, is there some ways that we can bring, and this is all coordinated with the Algae Foundation, and I'm on the board, I'm founding member of the board of that. So the question is there a way to bring the ATEC community in Texas together? Is there interest at Prairie View A&M to have some sort of algae focused initiative where we've already developed curriculum with DOE funding? We've had several $1 million. And one of the things we're supposed to do in the next year with DOE funding is expand our network. So we've reached out to Prairie View, among other universities and colleges and junior colleges. And, we're exploring opportunities to create collaborations and expand the ATEC network. There's about 10 or 12 schools and universities already, but like I said, quite a few of them in Texas. So, what's next? And so this is a first step dialogue, and happy to entertain that or pass the conversation off to the chair of the Algae Foundation, Ike Levine, who will be speaking to you guys in a few weeks. So I look forward to following up with all of you.
Thank you again. Thank you, everybody. Thank you, Dr. Mimby, for attending despite your busy schedule. And everybody, I'll see you next week. Have a good day.
Yeah, thanks again. Bye.
Bye-bye.